Okay, today I'm going to talk about DNA replication. DNA and RNA are nucleic acids, and nucleic acids are long biological macromolecules that consist of smaller molecules called nucleotides. In DNA and RNA, these nucleotides contain 4 nucleobases, sometimes called nitrogenous bases, or just simply bases, 2 pyridine and 2 pyrimidine bases each. One of the major differences between DNA and RNA is the sugar, with the 2-deoxyribose being replaced by the alternative pentose sugar in RNA. The 4 bases found in DNA are adenine, cytosine, guanine, and thymine. A 5th pyrimidine base called uracil usually takes the place of a thymine in RNA and differs from thymine by lacking a methyl group on its ring. DNA contains the genetic instructions used in the development and functioning of all living organisms. DNA provides living organisms with guidelines, genetic information in chromosomal DNA that help determine the nature of an organism's biology, how it will look and function based on the information passed down from former generations through reproduction. The slow, steady changes found in DNA over time, known as mutations, which can be destructive, neutral, or beneficial to an organism, are the core of the theory of evolution. DNA's genes are expressed or manifested through the proteins that its nucleotides produce with the help of RNA. Traits or phenotypes come from which proteins are made and which are turned on or off. The information found in DNA determines which traits are to be created, activated, or deactivated, while the various forms of RNA do the work. DNA is also found in the nucleus and mitochondria. It's a long polymer with deoxyribose and phosphate backbone and 4 different bases that I've mentioned previously: adenine, thymine, guanine, and cytosine. DNA structure is double-stranded. It has 2 nucleotide strands which consist of its phosphate group, 5-carbon sugar, and 4 nitrogen-containing nucleobases. DNA does not usually exist as a single molecule, but instead as a tightly associated pair of molecules. These long strands entwine like veins in the shape of a double helix. This arrangement of DNA strands is called antiparallel. The asymmetric ends of DNA strands are referred to as the 5' and 3' ends. During transcription, RNA, a single-stranded linear molecule, is formed. It is complementary to DNA, carrying out the tasks that DNA lists for it to do. Deoxyribose sugar in DNA is less reactive because of the carbon-hydrogen bonds. DNA lacks the 2' hydroxyl group and will not be hydrolyzed. Thus, it is a more stable polymer and better suited for storage of genetic information. It's also stable in alkaline conditions. DNA is also self-replicating. RNA helps carry out DNA's blueprint guidelines. It transfers genetic code needed for the creation of proteins from the nucleus to the ribosome. RNA has several different functions that, though all interconnected, vary slightly depending on the type. There are 3 main types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA transcribes genetic information from the DNA found in a cell's nucleus and then carries this information to the cell's cytoplasm and ribosome. Transfer RNA is found in a cell's cytoplasm and is closely related to messenger RNA as its helper. Transfer RNA literally transfers amino acids, the core components of proteins, to the messenger RNA in a ribosome. The ribosomal RNA is found in the cell's cytoplasm, and in the ribosome it takes messenger RNA and transfer RNA and translate the information. Depending on the type of RNA, the molecule is found in a cell's nucleus, its cytoplasm, and its ribosome. Like DNA, RNA is a polymer with a ribose and phosphate background and 4 different bases: adenine, guanine, cytosine, and uracil. RNA molecules are involved in protein synthesis and sometimes in the transmission of genetic information. The main job of RNA is to transfer the genetic code needed for the creation of proteins from the nucleus to the ribosome. This process prevents the DNA from having to leave the nucleus, so it stays safe. RNA is a single-stranded molecule. As a single-stranded molecule, RNA folds in on itself to link up its nucleobases, though not all become partnered. These subsequent 3-dimensional shapes, the most common of which is the hairpin loop, will help determine what role the RNA molecule is to play. The ribose sugar is more reactive because of the C-hydroxyl bonds. It's not stable in alkaline conditions and has larger grooves which make it easier to be attacked by enzymes. DNA is also replicated, but RNA is synthesized from DNA when needed. DNA replication is the process by which DNA makes a copy of itself during cell division. In molecular biology, DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. This process occurs in all living organisms and is the basis for biological inheritance. The first step in DNA replication is to unzip the double helix structure of DNA molecule as the hydrogen bonds between the bases are broken. This is carried out by the enzyme helicase, which breaks down the hydrogen bonds holding the complementary bases of DNA together, A with T, C with G. Once exposed, the sequence of bases on each of the separated strands serves as a template to guide the insertion of a complementary set of bases on the strand being synthesized. The new strands are assembled from deoxynuclease triphosphates. Each incoming nucleotide is covalently linked to the free 3' carbon atom on the pentose as the second and third phosphates are removed together as a molecule of pyrophosphate, or PPI. The separation of the two single strands of DNA creates a Y shape called a replication fork. The two separated strands will act as templates for making the new DNA strands. The nucleotides are assembled in the order that complements the order of bases on the strand and serves as a template. One of the strands is oriented in the 3' to 5' direction, or towards the replication fork, and this is called the leading strand. The other strand is oriented in the 5' to 3' direction, or away from the replication fork, and this is known as the lagging strand. As a result of their different orientations, the two strands are replicated differently. The leading strand is a short piece of RNA called a primer. It comes along and binds a short piece of RNA called a primer comes along and binds to the end of the leading strand. The primer acts as the starting point for the DNA synthesis. DNA polymerase binds to the leading strand and then walks along it, adding new complementary nucleotide bases to the strand of the DNA in the 5' to 3' direction. This sort of replication is called continuous. Now, the lagging strand, on the other hand, has numerous RNA primers are made by the primase enzyme and bind at various points along the lagging strand. Chunks of DNA, called Okazaki fragments, are then added to the lagging strand, also in the 5' 3' direction. This type of replication is known as discontinuous, as the Okazaki fragments will need to be joined up later. Once all of these bases are matched up, an enzyme called exonuclease strips away the primers. The gaps where the primers were are then filled by yet more complementary nucleotides. The new strand is proofread to make sure there are no mistakes in the new RNA sequence. And finally, an enzyme called DNA ligase seals up the sequence of DNA into two continuous double strands, and here is where the strands are combined. The result of the DNA replication is two DNA molecules consisting of one new and one old chain of nucleotides. This is why DNA replication is described as semi-conservative. Half of the chain is part of the original DNA molecule and half is brand new. Following replication, the DNA automatically winds up into a double helix. The two DNA molecules have been formed that are identical to each other and to the parent molecule. So why is DNA a more stable form than RNA? Well, DNA is a more stable storage form for genetic information. DNA is at least 100 times more stable than RNA. Removal of the 2-hydroxyl group from RNA to form DNA results in a backbone that is less susceptible to cleavage by hydrolysis and thus enables more stable storage of genetic information. In modern organisms, with the exception of some viruses, the RNA derivative DNA stores genetic information. The 2-hydroxyl group in the ribose unit of the RNA backbone is replaced by a hydrogen atom in the DNA. DNA also has a selective advantage over RNA as the genetic material. The genetic material must be extremely stable so that the sequence of information can be passed on from generation to generation without degradation. RNA itself can be a stable molecule because negative charges in the sugar phosphate backbone protect it from attack by hydroxy ions that would lead to hydrolytic cleavage. However, the 2-hydroxyl group makes the RNA susceptible to base-catalyzed hydrolysis. The removal of the 2-hydroxyl group from the ribose decreases the rate of hydrolysis by approximately 100-fold under neutral condition and perhaps even more under extreme conditions. Thus, the conversion of the genetic material from RNA into DNA would have substantially increased in its chemical stability. In all cases, the building blocks used in the synthesis of DNA are synthesized from the corresponding building blocks of RNA by the action of enzymes termed ribonuclease reductase. These enzymes convert ribonucleotides into deoxyribonucleotides. DNA is also more easily repaired. In both DNA and RNA, the base cytosine frequently undergoes a spontaneous chemical reaction known as deamination. The result of deamination is that cytosine changes into uracil. In RNA, which contains both uracil and cytosine bases, natural uracil bases and uracil bases that are results from deamination of cytosine are indistinguishable. Therefore, the cell cannot know whether the uracil should be there or not, making it impossible to repair cytosine deamination in RNA. DNA, however, contains thymine instead of uracil. The cell identifies all uracil bases in DNA as having been the result of cytosine deamination and can repair the DNA molecule. DNA's information is also better protected because the double helix structure of DNA places bases inside the structure, protecting the genetic information from chemical mutations, that is, from chemicals that react with the bases, potentially changing the genetic information. In single-stranded RNA, on the other hand, the bases are exposed and more vulnerable to reaction and degradation. The double strand also allows for double checking. When DNA is replicated, the new double-stranded DNA molecule contains one parent strand, which serves as the template for replication, and one daughter strand of newly synthesized DNA. If there is a base mismatch across the strands, as often happens after replication, the cell can identify the correct base from the parent DNA strand and repair it accordingly. I've talked about RNA being susceptible to hydrolysis. I just wanted to briefly mention what hydrolysis was right there, and here's the mechanism for RNA hydrolysis. I will go into it a little further here. RNA hydrolysis is a reaction in which a phosphodiester bond in the sugar phosphate backbone of RNA is broken, cleaving the RNA molecule. RNA is susceptible to this to this base catalyzed hydrolysis because the ribose sugar in RNA has a hydroxyl group at the 2' position. This feature makes RNA chemically unstable compared to DNA, which does not have this 2' hydroxyl group and thus not susceptible to base catalyzed hydrolysis. The mechanism, the RNA hydrolysis occurs when the deprotonated 2' hydroxyl group of the ribose, acting as the nucleophile, attacks the adjacent phosphorus in the phosphodiester bond of the sugar phosphate backbone of the RNA. There is a transition state where the phosphorus is bonded to 5 oxygen atoms. The phosphorus then detaches from the oxygen, connecting it to the adjacent sugar, resulting in ester cleavage of the RNA backbone. This mechanism is also referred to as RNA cleavage, and this produces a 2', 3' cyclic phosphate that can yield yet another 2' or 3' nucleic when hydrolyzed. There's other types of hydrolysis too. I just wanted to briefly mention. There's autohydrolysis and enzyme cleavage. But I wanted to conclude my presentation there. Thank you for listening, and I hope you enjoyed it.